Hello, everyone, and welcome to today's webinar, Keeping Up with the Chemist, Cloud Processing for Pharma CryoEM. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, visit them at thermofisher.com. Now, we encourage you to participate today by submitting any questions you might have during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speakers today. First, Taiana Maia, an Associate Director at AstraZeneca, and Steve Litster at AWS Principal HPC Business Leader for Healthcare and Lifesciences. Joining these two will be Brian Skjerven, an AWS Specialist Solutions Architect, and Natalie White, AWS Enterprise Solutions Architect. And finally, Ieva Drulyte, a cryo-EM application scientist at Thermo Fisher Scientific. Welcome to all of you. You may now begin your presentations.
Hi, everybody. I'm Taiana Maia de Oliveira, and I'm heading the cryo-EM group at AstraZeneca in Cambridge, UK. I'm very happy to be here today, so many thanks to the organizers for the invitation. In this talk, I will elaborate a bit on how structural biology and cryo-EM fits into the drug discovery process and how important it is to have structures at the right stage of the process. At the final part of the talk, I will also share some of the experience in implementing and expanding the use of cryo-EM at AstraZeneca and compare the state of things in the beginning to how they are now. So, as you know, drug discovery is a lengthy, complex, and costly process. Developing a new drug can actually take 12 to 15 years. On top of that, this process is permeated with a high degree of uncertainty that a compound will actually succeed as a drug. The success rate for candidates is around 10%. So in this slide, what we have is an overview of the different stages of drug development process until the approval and post-market surveillance. This diagram also shows some of the specific concerns and some steps that each stage prioritizes. So, for example, we have in target identification, we need to find out the disease linkage, the druggability, and the validation of the target. So we are focusing here on structural biology and therefore cryo-EM. And I want to tell you that the stages in which cryo-EM and structural biology have the greatest impact is really in the early stages of the process of drug discovery. So as you can see in the slide, these would be the phases of target identification, hit identification, lead identification, and optimization. The uses of structural biology, they will include, for example, the assessment of druggability of the target in question, and also that happening at the target identification stage. The final validation of the compounds that were identified through the screening campaign, they can also be done by structural biology, and they are done at the stage of hit identification. However, we will focus a little bit on the stage that has the most extensive use of structural biology, and that use is really optimizing a compound into a molecule that has drug-like properties in the lead optimization stage. So in this slide, what I want to do is really illustrate this process and how we use the structures. Also, after I present this, I want you to have a feeling for how iterative is the nature of this process. So in this case here, we have a hit in the first box, which you can see, and we managed to get a structure from the compound in complex with the protein of interest. And what you can see with the arrow is that there is an empty space on the pocket right next to the compound. Now, this was a compound that was picked up during the hit identification stage through screening. And it has very poor potency, so you can see the IC50 is around 5 micromolar. Now, having the information from the structure and seeing that there is this empty space, there was something obvious to do there, which was to try to optimize the stage into getting a better shape complementarity. So for this, we introduced a phenyl group that could really fulfill that pocket properly. Now, what is usual with this process is that when you solve one problem, you get another one. And in this case, by adding that group, we now have a compound that is has very low solubility. Now, the structural information can also help there, because from this structure in the middle, we can see that there is plenty of space to grow a compound at the edge of the pocket. So the chemists can then exploit this information to see where they can put a solubility group to improve the physical chemical properties of these molecules. And this was exactly what was done in this case here. So in the previous slide, it's a very specific example, and now I would like to go for a more general description of how the process actually goes. So the project team repeats a common workflow to optimize the identified hits toward clinical candidates. This is called the DMTA cycle. Everybody knows that Pharma loves acronyms. This is the first one, DMTA. And DMTA means design, make, test, analyze. What exactly does that mean? So if you look at the diagram, what we can see is that in the left, we have the hit, which came from the screening cascade. And this is the chemical starting point for a drug. Usually, this chemical starting point has several properties which are not very attractive. It might be weak, it might be poorly selective, it might have poor properties, and it might be toxic. And what you want to do during this cycle is design novel chemistry, that's the design step, synthesize compounds, that's the make on the DMTA, put those compounds through assays, and determine structures, that is, if the structural support is enabled for this project. And then you analyze that and construct a predictive model. That predictive model will tell you how to make, what to modify in the compounds to start over the cycle. Hopefully, in the end of the cycle, what we get is a candidate, which ideally is potent, selective, effective in vivo, stable, and safe. So one of the problems here is that to really get a good predictive model, you need to synthesize a lot of compounds. The structural biology support is really good because it can decrease the number of compounds needed to actually get a qualitative predictive model at an earlier stage. So we can cut, for example, synthesizing 10,000 compounds to maybe synthesizing 1/10 of that. The other consideration that we have to do for the structural biology support is that it's great to have a structure, but it needs to be at the right time because the DMTA cycles are under constant pressure to be sped up. So let's go a little bit more into the cryo-EM since I set the stage for you to know what's the impact that structural biology can have on the structure-based drug design and on the clinical candidates. So everybody was hugely excited about the possibility of having previous intractable targets, especially membrane proteins, enabled to structural biology by cryo-EM. But there was, of course, a lot of skepticism. And part of that was relating to the question of, could we get high-resolution structures in the timelines that were suitable to inform the project? To answer that, I will start a bit with the cryo-EM experience that AstraZeneca had. So AstraZeneca joined the Cambridge Thermo Fisher Pharma cryo-EM consortium back in 2016. This is a partnership among the MRC LMB, the University of Cambridge, Thermo Fisher, Astex, AstraZeneca, GSK, Heptares, and UCB. We had access, together with these four other companies, to a cryos that was housed at the University of Cambridge. So our structure writing, we realized right in the beginning that many of the high-resolution structures required really large data sets of 5,000 to 15,000 images. And especially at the beginning, the microscope time and the processing time that it was required to actually get to these structures really limited the number of structures that we could generate. So at that stage, what we had more impact on was really on the final heat validation and characterization phase. So if you remember back to the diagram that I showed, this basically meant that we could know the binding mode, and we, instead of going through several iterations, we could try to get some of that information through computational methods. Also, right in the beginning, very early on, we faced two obstacles that had to do with the computation and the processing front. The number one was the fact that achieving an optimal cluster computing setup with a centralized IT department was not trivial. So a solution for that was actually to find an alternative, and our alternative was to invest in a software that was less reliant on parallel computing. So we started a partnership with CryoSPARC last year. The second was the fact that transferring huge amounts of data to our processing server was very time consuming. It was all based on disks and you know, transferring from the microscope to the disk, and from the disk to a computer, and then to the central server. So we actually adapted that into a workflow with a transfer through AWS, and that actually works really well, and it can go the transfer can go right by the side of the data collection. So our experience now is quite different from the start, from all of this that I've been telling you through this slide. That's for several reasons. First, some solutions I already told you about, but there were several improvements on other factors here. One was the improvement in the camera throughput, which has been extremely impressive. We now usually collect images 120 to 250 images per hour. We got increased microscope access. This was increased when the consortium invested in the second cryos in 2018. And we had several improvements on EPU and on the ease of data collection. And that, of course, has an impact because it frees up staff time for structural determination and model building and interpretation. So this is the current state of affairs of CryoEM and AstraZeneca, just to give you an idea of where you're standing right now. So at the current state, what we have is 5 projects with cryo-EM systems in place to generate co-structures for chemical equity optimization. So we can really have an impact on the lead optimization stage, and we can really have several structures and really inform very thoroughly the process. This is because dozens of structures per project are possible with the setup that we have, and that really allows to build a structural activity relationship model. So if you remember the predictive model building on the DMTA cycle, this is what we are enabling now with having all these structures. So just for illustration, in an optimal case, we can go from a pre-optimized system. We can have data collection for 1 day, concomitant transfer, and then 2 days to obtain the map, and then another 1 to 2 days to build the structure. So that takes us from sample to PDB in 5 days. This is, of course, the best case scenario. The average case is that we get structures for one project every second or third week. And this is because of several bottlenecks. The processing sometimes can take several rounds of 2D and 3D classification. That depends a lot on the system. Some of them even take a very thorough 3D variability analysis to get a high quality structure. Sometimes we have a lot of protein movement, so the building is much more complicated than thought of, and then you actually spend quite a lot of time at that part. And the other factor here is, of course, the scientist workload, because we are supporting several projects at the same time, and we also have to cope with data collection at the microscope. So on my final slide, I just really wanted to make the point that CryoEM is delivering the promise, is really bringing previously intractable targets into the realm of structure-based drug design, and this is not science fiction anymore. So I would like to thank you for your time, and I'm eager to hear some questions in the end, but now I will give the word to Brian.
Thank you, Taiana. So I'm Brian Skjerven.
Brian, I think I'm in control still.
Oh, there we go. Yeah. There we go. All right, thank you. Thanks, Taiana. So I'm Brian Skjerven. I'm a solutions architect at AWS. My focus is on high performance computing, and I'm going to talk about some of the work that myself and my colleagues Natalie and Steve have been doing in the space of cryo-EM on AWS. And so this started sort of last year when Structure Biotechnology, who was a partner with AWS, came to us and wanted to know, what would CryoSPARC look like on AWS? How do we set this up? What's sort of the best practices? Help us out with this. And so we proceeded to start to walk through sort of what their requirements are and build a reference architecture around that. And a few things came out of the CryoSPARC requirements. The first one is obviously access to GPUs. And this was a bit of an interesting problem because in some cases, stages in CryoSPARC scale well. They scale over multiple GPUs, and other stages are single GPU only. And so that's there's a need for some sort of flexibility in the compute resources available. The other point was around storage. So typically, Structure has recommended that you have some sort of fast local storage on the compute nodes to enable, have a cache to keep those GPUs fed with cryo-EM data. And so we have compute resources available on AWS that can address that, but not all of our EC2 instances have that fast local storage. And so there is a question around, do you always need to use a specific instance type, or can we mix and match different storage options? And the answer is yes, and I'll get to that in a second. And then there's also some questions around, can we do this in a cluster environment with single or multiple users? And so what we came up with was basically a reference architecture built around a couple of AWS services, particularly something called AWS Parallel Cluster and Amazon FSx for Lustre, which I'll go through in a second. And we published an architecture with Structure, as well as a set of benchmarks and cost analysis. So very quickly, I'll go through some of the core services that make up this cryo-EM solution on AWS. So the first one is AWS Parallel Cluster, which is a way to easily create, configure, and manage HPC clusters on demand. It's a single command line option, and you can spin up a cluster from a single configuration file in about 15 minutes. And so it handles all of the sort of heavy lifting that you would normally do to create a cluster, things like setting up a job scheduler, setting up queues, doing things like shared file systems. All of this is handled by Parallel Cluster, and so you basically just need to pick certain options and press go, and it creates your cluster for you on demand. It can also do things like scale up your compute nodes when you submit jobs, and then scale them down when you don't need them. So you're really only paying for the compute that you use. And so all of that is built on Amazon EC2, which is our Elastic Compute Cloud. That's our range of different instance types, and I'll get into some specifics on that in a little bit. I mention it here only because it does make up our larger HPC services on AWS, but we have something called Elastic Fabric Adapter. This is a high-speed interconnect, low latency, high bandwidth interconnect to enable these sort of tightly coupled MPI type jobs that need to run as a group on a collection of nodes. That doesn't necessarily apply to cryo, or at least to CryoSPARC, but we are investigating other applications like Relion, where this can be made use of. And then the other key part to this is Amazon FSx for Lustre. So if you're not familiar with Lustre, it's a high-performance parallel file system. It's fairly standard on most HPC clusters around the world, and we offer a fully managed service for this. So it's basically you point it to a S3 bucket of data and select a size, and you press go. It integrates very nicely with AWS Parallel Cluster, and I'll show you some of that in a little bit. And then one other part to the HPC services on AWS is something called AWS Batch. It's sort of the opposite of Parallel Cluster, where Parallel Cluster is designed for these tightly coupled MPI type jobs. AWS Batch would be used for sort of large independent parameter type jobs. And so moving on. So I just want to highlight a little bit about some of the EC2 instances that we made use of. We have a really broad and deep platform of instance types, over 400 types that can be used for everything from machine learning to big data to HPC. And this was a question that Structure had. What do we use? Because not only do we have we've got GPU options, we have CPU options, but they also come in a range of different sizes, different memory configurations, different network configurations, and so the question was what's going to work best on AWS for CryoSPARC, and the answer was a little bit surprising. It was, and I'll show you the benchmarks later. Is the best approach is to use a mix of GPU instance types for CryoSPARC. you're not locked into one single instance type, like you would be in a in a workstation or an on-prem cluster where you procure hardware and you're using that for a couple of years before you upgrade. With parallel cluster, we can easily switch between low cost NVIDIA T4 GPUs, or if you need, you can switch to a larger latest generation NVIDIA A100 GPU, and you can do that all within the same cluster. And so I'll walk through in the benchmarks a little bit more of the specific GPU types. And so one of the other, as I said, beyond just the question about compute resources, Structure also asks us about data, and this is a problem that we see with a lot of customers in the cryo-EM space, and that is just managing data. You produce massive amounts, multiple terabytes of data. How do you organize that? How do you get it to your compute resources? And in the past, there's been a few ways that people have done this. You can put it in an S3 bucket, and then you may have to copy it to like a local storage, local disk attached to your instance. Maybe you're creating your own file systems, like setting up your own shared file system, or you might just be doing something like running it directly from S3, which simplifies things, but you're still having to run over a network connection, and there is a delay, there is a performance hit from doing that. And so this is where FSX for Lustre comes in, because it simplifies this process. As I said, it's a fully managed high-performance parallel file system. It connects to an S3 bucket, and so the idea is you upload data to an S3 bucket. It can automatically be brought in to this high-performance Lustre file system, which is basically acting as a high-speed front-end cache for that S3 bucket. It can give you access to hundreds of gigabytes of throughput. Basically, for every terabyte of storage, you get another 200 megabytes per second of throughput. We can do millions of operations on it. And again, the other key is this is all managed. It basically is pointed to an S3 bucket where your data is, and press go. And there were some questions around, how would this perform versus the traditional approach that Structure had gone with of using local file systems. And so we did. This is one of the benchmarks that we tested. So what you're looking at is basically Lustre performance versus local drives on our instances. And this is the percentage decrease in runtime. So higher is better in this case. And so in nearly every case in this 2D classification step, and we chose this 2D classification step particularly for this because it can use a cache feature in CryoSPARC, and it can scale across multiple GPUs. So we wanted to see, what was the effect if we were running on a single GPU or multiple GPUs. And, we're upwards of over 50% performance improvement in some cases with this. And I'm happy to get into some of the technical details later in the questions with this, but the shorter answer is FSX for Lustre allows us to decouple storage from compute. We don't have to pick compute types that have fast local storage. We can go with lower cost single GPU types if that's what's required, pair it with a high performance file system, and still get good performance, as opposed to having to lock into a single large instance type that has that fast local storage. So this was actually a pretty game changer for Structure and what they recommend now. The one thing I want to highlight, to just talk a little bit about what Taiana had mentioned about problems with data management. One of the great features with FSX for Lustre is this auto import feature. So you can basically use S3 as a big data lake. You're uploading data there, and you can automatically bring data into your Lustre file system. And you have a lot of control over what gets automatically brought in. That can be data that exists only in the bucket when you create the file system. You can bring in new objects. So you could imagine a case where you are continually uploading data from your cryo-EM microscope into a bucket. It hits the bucket, it's automatically brought into a high-performance file system that's attached to your compute nodes, and you're running something like CryoSPARC Live in that fashion. Or you could even upload modified data. So if you had to rerun experiments, upload new micrographs, process them, that's an option too. So this managed auto import feature can really simplify some of these data management problems that people are facing. And so when we put all this together, FSX for Lustre and parallel cluster, we came up with this architecture, and we wanted it to be pretty straightforward and easy to use. The idea behind it was it will look and feel much like an HPC cluster that users are used to, but with, again, it's all managed and easy to set up, and removes a lot of the heavy lifting and sysadmining that you would have to do. So we built this with the idea that you'd have multiple compute options. So we have multiple queues here that run a range of different NVIDIA GPU types. As I mentioned, we can run the latest generation A100 GPUs, V100s, and T4 GPUs. And I've got benchmark data where you'll see why having this flexibility is important. We've also, you can also add in traditional CPUs like our Intel Skylake options to run on things like Relion. We've got a number of shared file systems here. The one I highlight in the diagram is for the cryo-EM data. This is our Lustre file system, but there are also a few others that are more of your standard network file systems. This is for things like a shared home directory and application install, like where CryoSPARC is installed. And the other thing I really want to highlight here is that this is all designed to be created and brought down with a single command line option. It is infrastructure as code. You can manage multiple clusters from single configuration files. You can bring them up when you need. You can tear them down when you need. You can automate their deployment. You can automate file system creation. The idea was that this was meant to be flexible and that you would only pay for the compute and the storage that you need, as opposed to having to have a HPC cluster up all the time. And sorry, the color is wrong in the top part there. This was the benchmark that we did. This was the Empire 10288. This was a cannabinoid receptor. It's a fairly small benchmark. We did this in part on structure as recommendation, but also because we could run a large number of simulations and so to get a lot of benchmark data. You can see the pipeline that I ran there. These were all the steps, and I ran this on anywhere from 1 to 8 GPUs, and I ran this on all the GPU types that I mentioned there, the NVIDIA A100s, V100s, and T4s. And I'll say there's the full benchmark data is available on CryoSPARC's website. They have a section on there called CryoSPARC on AWS. The whole reference architecture that we built for them is there, along with a lot more of the benchmark data. Basically, I give breakdowns of the performance of each stage of the pipeline. But for brevity's sake, I'm going to just show you a couple of slides. By running a lot of these different simulations, we got to see a view of what a cryo-EM pipeline looks like on AWS, because it's not like a lot of other HPC applications where you're running one single code and you need to see the performance for that one application over a range of over a range of CPU count. This is, you've got multiple stages. Each stage is going to behave differently. Some scale over GPUs, some don't. Some are more affected by file systems than others. And so what we were able to do is put together graphs like this, where we see how different stages scale as we run on more GPUs, or as we add in something like FSX for Lustre. And to sort of distill, I've kind of distilled it down to 3 points there, where we saw certain stages scale really well, like patch motion and patch CTF. These are ones where you would want to run on a larger 4 or 8 GPU option. There are other stages like 2D classification or extraction, where running on a single GPU to save money is sufficient. You aren't gaining a lot by running on multiple GPUs. And then there are others where, like ab initio reconstruction and non-uniform refinement, where these are single GPU stages, and so your decision point there really comes down to cost. Do we pay for a newer, larger GPU and get a faster time to solution, or do we want to save cost and go with like a lower spec NVIDIA T4 or something like that? And so by running all these different benchmarks, we can start to put together different cost analysis. And I've got 2 examples there. This is by no means set in stone, but it's just meant to illustrate this idea that when you mix and match different EC2 instance types, different compute resources to the different stages of the pipeline, you can work out a very fast time to solution while still keeping costs down. So the chart on the left just sort of shows what the total cost, including storage, what the total cost of each of those simulations would be if I ran it on a single instance type. So for example, I'll pick one, the red. If I were to run on 4 GPUs on this P3DN, which is a NVIDIA V100 GPU, I'm at $128, and this was around 3.5 hours of runtime. But I've got other options where like in the chart on the table on the right, where if I mix and match and do a little bit of benchmarking, I can run on cheaper instances. I can bring in some cases get my time to solution lower and still save on cost. And so this was really exciting for us. We published this. We've had a lot of interest in this, but what we really wanted to see was what does it look like on production data? Because as I mentioned, this was a pretty small data set, and so this is where Thermo Fisher came in. And so I'm going to turn it over to Ieva, because Ieva actually took what we built and actually started to use it on real production data, which is exciting for us. And so with that, I will say thank you, and I'll table any questions for at the end, and I'll hand it over to Ieva.
Thank you, Brian. And hi, everyone. My name is Ieva, and I'm an application scientist at Thermo Fisher Scientific. So, like everyone else, we're really excited to see these benchmarks, and we wanted to see how the technology works in our own hands, and especially on big data sets. So today I will show you 3 benchmarks in total. Two public data sets, which are available on Empire, and 1 data set from Thermo Fisher. The first benchmark I want to talk about is calcitonin gene-related peptide receptor bound to its agonist. So it is a small membrane protein, 74 kilodaltons, and such small size proteins are traditionally more difficult to process because they are small, they have less signal, and they might require more data or more processing time to arrive at the solution. So we thought it's a great idea to start with this benchmark. Downloaded the data from Empire, and the data set contained 5.6 thousand movies, from which I picked 6.3 million particles. And in total, it took me 4 working days to arrive at the solution. And that's because, well, it was the first time I saw this data or worked with this protein, had to think about the right steps, and didn't work through the night. So yeah, it took 4 days. But if you look at the time, GPU clock time that it took to write the structure was 36 hours. And the final mass is 2.8 angstroms, and it consists of 1.4 million particles. So on the right, you see 2 maps. The one in gray is from EM database, and the one on the right is from the AWS benchmark. And you can see there is definitely improvement in resolution, going from the published structure, which I would say is mainly attributed by non-uniform refinement from CryoSPARC, because it can really deal well with small membrane proteins in my experience. So all processing was done in CryoSPARC on AWS on a P4 instance using A100 GPUs. So this was not the type of work that Brian described, where we just ran a lot of simulations. We didn't choose that path. So the second benchmark is also a GPCR, but this time a lot larger complex. So it's an active glucagon-like peptide 1 receptor bound to GLP-1 and a G protein. And similarly to the first benchmark, we downloaded the data from Empire, 5.7 thousand movies, from which I picked nearly 5 million particles. And similarly to the first example, it took me 4 working days to arrive at the final map, or in other calculation, 38 GPU hours. So the final map, the shown in blue, was 2.7 angstroms, containing 1 million particles, and you can see it's fairly similar to the published map showed in gray. And for the final benchmark I want to show you, which shows a soluble protein that many of you will recognize. So it's a SARS-CoV-2 Spike in complex with a Fab. This was a collaboration with Chemical, who developed an anti-spike antibody and sent us a Fab derived from that antibody, and we did a structure of that in Thermo Fisher. So for this case, we collected the data in Falcon 4 using ER mode, 2,500 movies, and on-the-fly motion correction was performed alongside data collection. And we were testing a different case here. We just uploaded the motion corrected dose weight micrographs to Amazon. And this was really fast. And then I could pick 440,000 particles from this data set. And it took me just half a day of work, of submitting different jobs to CryoSPARC to arrive at the final map. And the GPU hours accounted to six hours in total from start to finish here. You can see the 2.9 angstrom map of the spike core on the right. It contains 64,000 particles. And if you contour it to lower level, you can also see that the Fabs can be identified bound to the open conformation of the receptor binding domain circled in red. So next slide, I want to show you a breakdown of all processing times in CryoSPARC. And in these three examples that I showed, the total runtime was anywhere between 5 hours, 40 minutes, and 1.5 days. And if we look at the sort of graphical representation on the right, you can see that pre-processing steps and 2D classification actually took the least amount of time, and the most time was spent on 3D classification methods. So I pulled together heterogeneous refinement and initial reconstruction in this category, which I both used to select the best GPCR particles for reconstruction. So I do believe there is definitely some way to make the process even more efficient, the data processing workflow more efficient, and with some optimization, the repeat structures of average or difficult cases may be computed within 1 to 2 days. Okay, and so that brings me to my last slide, and I want to share that I've been using CryoSPARC on AWS for some weeks now, and I'm really enjoying it, especially because, as Brian said, you have a choice of so many different GPU instances, and you don't have the same bottlenecks that you may have with data storage and so on. So it's for sure faster than some of the in-house solutions I've seen or worked with. So here are my conclusions. AWS Parallel Cluster and FSX for Lustre definitely offer the scalability and fast performance for cryo data processing. And together with CryoSPARC, it allows you to calculate sub-3 angstrom structures of large data sets within hours to days. I would say definitely not weeks. And that means that cryo structures can be turned around fast enough to be in sync with medicinal chemistry cycles and actually bring value to structure-based drug discovery platforms. So I would like to thank all the people without whom it would not have been possible to do this work. So Ali and Sarah from Structura and Zara from Chemical. All people from AWS, so Brian, Natalie, and Stephen, who really guided us through our parallel cluster setup and all of the steps to get started. And of course, I'd like to thank my colleagues at Thermo Fisher Scientific, especially Adrian Co. and local and central IT teams who helped with the data transfer and all other IT parts on our side. So thank you very much for your time, and I believe it is now time for questions.
And I want to thank all of you for those informative presentations. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. So welcome back, all of you. And we have a couple other people joining us today. We have some great questions coming in already. So let's start with our first question. What resolution do you need for lead optimization?
I guess that's for me.
And any of you can take these questions. It looks like, yes, this one's directed at you, Taiana.
Yeah, so again, we have to be a bit careful with the average resolution because sometimes that's not very significant. But looking at local resolution, I would say between 3.3 and higher than that, you should be fine.
Wonderful, thank you so much. And how do you decide how on the number of rounds for heterogeneous refinements?
I can take this one. So I don't think there's any rules there. And I might also not be doing it the perfect way, but I use heterogeneous refinement as fast 3D classification to get rid of obvious drug particles. So I ask the CryoSPARC to divide my particles into a good class and 1 or 2 bad classes, and I see how many particles in each round I'm throwing out. When that number becomes very small, then I'm confident that the stack of particles is good, and then I go forward.
Thank you so much. Now, Brian, this question's for you. Does AWS support Docker containers? And go ahead.
Yeah, so broad answer is yes. There's some specifics in there. In parallel cluster, yeah, we can run containers. We generally recommend using Singularity, and you're welcome to ask a follow-up question on that if you're not familiar with it. That's mainly because it will simplify some of the things around running Docker in a cluster. The other option is AWS Batch that I alluded to earlier. It is designed entirely to work with containers. We tend to see people use Batch less for cryo-EM only because they don't really have this need of doing large, embarrassingly parallel applications. They usually need to do, like Ieva did, one kind of a couple of large intense runs, and they need to have some sort of interactive approach to it. But in both cases, yes, we can run containers.
Wonderful, thank you so much. And I want to thank our audience for these great questions coming in. Our next question, Ieva, what software did you use for the on-the-fly motion correction of the third data set prior for uploading to AWS?
Thanks. I should have mentioned that in the presentation. I use CryoSPARC Live, which is also a great piece of software to do on-the-fly analysis of the data coming off the microscope. So I used that, and that was run on just a local machine with 4 GPUs.
Thank you so much. And one more follow-up question for you is, Ieva, the computing process from frame alignment to final high-resolution map, is it completely automated?
Sorry, can you repeat the question?
Sure. So, is the computer process? It says the audience member says, "Hi Ieva, the computing process from frame alignment to final high-resolution map, is it completely automated?"
Yeah. Yeah. I wish that would be the case, but there are definitely advancements coming in, but there are decisions to be made there, and at every step that Brian described in his pipeline and that I showed, that you have to make decisions which particles to take to the next step and how to tune it. But I think, especially in the case of routine analysis, like repeat structures, there are definitely some ways to automate the process and to streamline it to make it more efficient.
Sorry, Ieva, can I just add a little follow-up to that? Yeah, as Ieva said, we really can't automate around the interactive parts, but for the benchmark, I ran probably close to 200 different simulations, and the way you work in CryoSPARC on parallel cluster is you can create the next jobs that you want to run, link the output from the previous step, and CryoSPARC will submit that, and it submits a job to the scheduler on parallel cluster, which will provision a node, run the job, and then tear it down. So as long as you know what parameters you need for the next step, and it's nothing that has to be interactive, you can automate that to some extent, and parallel cluster will automate provisioning the compute resources. But I'm not a cryo-EM scientist, so I can't advise you on what parameters you need to add in. You'd have to do that as well.
And thank you so much for adding that information. And Taiana, this question is for you. Do you use cryo-EM for biologics?
Yes, we do. We use it in a less intense way than we use for small molecules. So usually when you're trying to optimize a small molecule, you really need several structures of the follow-up compounds. And in case of biologics, antibodies, and things like that, the affinity maturation is usually very good already. So it only comes to structural biology support within AstraZeneca when there's something that quite doesn't add up. So if there's a question, if the antibody is not behaving in the expected way from the assays, then and is a molecule of interest, then it comes to us, but it's not as iterative as for the small molecules.
Thank you so much. And again, I want to thank our audience. We have so many great questions coming in for our presenters. How about transfer costs? Are there any transfer costs for uploading data to AWS?
I can take that one. So in general, data transfer into the cloud does not have a cost associated with it unless you're using one of our more managed services to do that transfer. But if you're directly transferring data into S3, there's a storage cost associated with it, but the actual data transfer itself doesn't have any additional cost associated with it. And what's great is once you have the data in the cloud, you can use all of those cloud native services or any additional applications or software that you want to install on your cloud infrastructure once the data is already there.
Wonderful, thank you so much. And Brian, this question is for you. Is a new parallel cluster created for each separate test? And how big is the overhead of this creation, deletion, and or cost? And they also want to thank you for your presentation.
Thanks. The answer is to your first question about a separate cluster. It depends. What I did in this case was create a single cluster and run multiple jobs on that. You could create individual clusters if you wanted, if you had a need for that. Maybe you have different data sets, different groups of users that you do need to keep separate. You could do that. I mean, that's the great thing about how this is designed. It's designed to be brought up, used, and created on demand, and so you can do that once for a lot of people, or do a lot of little ones for individual users. In terms of the overhead, probably the biggest overhead comes in the file system creation. So creating the Lustre file system involves basically setting up the back end of some EC2 instances that are going to manage this Lustre file system for you, and then also some of the data import. And in general, that's around 15 minutes. It kind of scales a little bit depending on how big of a file system you're doing, but that's probably the largest overhead. In terms of cost, really all the you need at the bare minimum is this head node that you log into, and that's where you interact with the CryoSPARC web interface and submit jobs. You can create a cluster that has 0 running compute instances at the start, so you don't need to have those up and running until you're ready to use it. So from a cost standpoint, the initial cost on parallel clusters can be very low. There's a lot of options that we can tweak on that, and I'm happy to talk with that offline. But the initial overhead cost is pretty low. In some of the work that Ieva and her colleagues and I, in doing this, we must have created 12, 15 different clusters, and in some cases we were tearing them down, rebuilding them, tearing them down, rebuilding them in multiple times in an hour phone call. So it is really that easy to do it.
That's what's great about the solution is you can just configure it just from a text file, how many compute nodes you need, what the instance sizes need to be. It's very flexible.
Wonderful. Thank you very much. Now, what does the $ prices mean on the one slide? Someone is asking.
I'm going to assume this is about the cost on the breakdown that I gave. That's the total cost of what your compute resources and your storage for that single simulation. And so we basically just looked at what was the runtime for this entire simulation, what's the price per hour of that instance, and that's how we come up with it along with storage. So it's just to highlight this pay as you only pay for what you use approach.
Thank you so much. Now we have time for one more question so that I can allow you all to have some closing remarks as well. Are there any plans to provide a public hosted managed CryoSPARC version so you wouldn't have to deploy it yourself?
Well, just checking if one of the other colleagues wanted to. Steve?
Yeah, I'm happy to have that. Actually, that's an ongoing discussion that's more sort of directly related to sort of the CryoSPARC folks, but we are discussing that with them as well. So we'll follow up with you on that. But really, that's the structure biotechnology folks as well.
Wonderful, thank you. And I want to thank all of you for your presentations today. This has been a wonderful presentation. Would you like to provide any closing remarks? I'm not sure if all of you want to, or how about if I just allow any of you to just say anything if you'd like to add before we close?
I'll jump in and just talk about the partnership between all of the AWS people and Thermo Fisher and AstraZeneca. I think sometimes if you haven't ever run anything in the cloud, it can be a little bit overwhelming seeing all of the technology and the acronyms and the instance types and the costs. And I just want to reassure attendees that AWS has resources and people to help you with that. We've worked very closely with Thermo Fisher to make sure that Brian is obviously focusing on the HPC side, but working with Thermo Fisher's IT personnel, making sure that all of the networking and data transfer is happening in a cost-optimized and secure way. We have a lot of best practices, resources that are publicly available, training that's publicly available, demos and videos that are publicly available, so you can start your learning journey wherever you are and start building with the skills that you already have and kind of learn new skills as you go. So yeah, the success that we've seen on this webinar has just been phenomenal, and it's been really great to talk about the results and see the significant acceleration of the science that's happening at Thermo Fisher and AstraZeneca. So thanks everybody for all of the partnership that we've had in the past few months and bringing this to light.
You're welcome. And did anyone else want to comment? Okay, I want to thank all of you again for your time today. Taiana, Steve, Brian, Natalie, Ieva, all of you, thank you so much for your research and for your important work. And I also want to take time to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information we provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, stay safe, everyone. Take care. Bye-bye.